The second problem I was going to talk a little bit about today, just to give you, just so we've spent some live time in the classroom talking about how to use this DESeq2 stuff. So I'm going to go to ngs.r and we'll look at edgeR and we'll look at DESeq2. Both of these are statistical methods for doing hypothesis testing, differential expression analysis on next-gen sequencing data, specifically RNA-seq data, where the sequencing technology has been used so that we can quantify counts of RNA copies of different genes. So we're quantifying activity levels of genes, but we happen to be using sequencing data, next-gen sequencing. One of the issues here is what we've talked about with the nature of the data that we see being counts, read counts. And that presents some statistical challenges because, for example, t-tests and linear regression are not geared to work with counts, which can mean the p-values and the false discovery rate estimates you get if you were to use a t-test could be incorrect. What people generally do, as we've talked about, is treat read counts as essentially like Poisson distributed data. That's natural to think about because Poisson is appropriate to counts. But the issue with using a Poisson is that the read counts themselves don't generally really follow a Poisson distribution. Just because they're counts doesn't mean Poisson is the right distribution, because the Poisson has some a relationship that it assumes, namely that the variance is equal to the mean. And that's not generally the case with read count data. So edgeR and DESeq2, both of them do something very similar, which is to say, let's assume a Poisson model on our read counts, but let's allow for a more flexible Poisson model than the default version, which says that the variance has to be equal to the mean. Okay, so it's basically Poisson distributed data with a twist. And that's why we need specialized methods, because it's not just t.test that will do this for us. edgeR, DESeq2, they both do this task. Let's analyze our read count data with overdispersed Poisson distributions. They come from different author teams, so they operate a little bit differently, and we'll see how they operate a little differently, but they're both trying to do the same thing. Let's start with edgeR. So here, line 232 in ngs.r is example with edgeR. We need a couple of packages from Bioconductor. We need edgeR and we need GoSeq. We might already have GoSeq, I'm not sure, but we'll check. So I'm going to get in this function that we've used before to install Bioconductor packages. And then let's come back down to where we were and library or install these two packages that we need, edgeR and GoSeq. Yeah. So this takes a moment. Once we get this installed, we're going to use an example data set that comes with edgeR to illustrate it. So let's see these data. So LinCap is the name that we'll give it. And here's a description. So these are data that examine the effects of androgen stimulation on human prostate cancer cells. So we have human prostate cancer samples, and you can isolate the cells from that tissue sample, and you can maintain them, keep them alive indefinitely so that you can study them. We have cells like that. We have 17 million reads, so a huge pile of reads that have been obtained from the NGS instrument on a collection of individuals. Okay. And then we'll read in the data as soon as we get the package installed. Okay, let's update everybody. This came out of the help file for EdgeR. This is just going to get the actual data. So sometimes when you install a package from R, the package will come with a data set. We've seen that before. We'll library in a package, and then we'll say data set name, and it will load in the data. This is another way to access a data set that comes with a package. If you install a package, then somewhere on your computer there will be a folder created for that package. And inside that folder might be additional folders where there's data sets. So you don't have to know how to do this particular command. It's kind of cryptic, but what we're doing is reading in data that came with EdgeR. Let's see how we're doing back here. So we'll read these in, and then just like with your data from the homework, the hammer data, there's a bunch of rows in the read count table that are all 0. What does that mean? That each row in the data set that we read in with these next-gen sequencing data, each row corresponds to, for our purposes, a gene. So some location in the genome that has been identified as a coding location, a coding region. And then in the cells for that row will be counts for the fourth person in our fourth sample of these cell types, this cell line. The count for gene 12 is equal to such and such. That's the number that would be in row 12, column 4. Some of the rows have all zeros. So that means that while that gene that the row corresponds to is known in humans, we know there's a gene at this location, we did not see any reads that were aligned back to that location. All right. So we are going to filter out cases where that happened. Genes for which there is no reads, we're going to get rid of it. And R is not responding. Taking a while. Okay. Once we've filtered out the 0 count genes, we have to create an object of a special class for both EdgeR and DESeq2. They require that you put your data, your read counts, into a specific kind of organization that it recognizes. We've seen this before also, that often when we have these external packages, we have to work with an object of a special class. And here's EdgeR's object of a special class. It's called DGE list. DGE, I think, stands for digital gene expression or something like that. Digital as in 0, 1, present absence. That's where this digital stuff comes from. If you read these papers, they're talking about presence, absence kinds of data. That's where read counts come from. We're going to pass in our matrix of read counts. We're going to pass in what EdgeR calls a library size. So if you deal with next-gen sequencing data, you'll hear this library jargon. You'll hear, here's my library. Here's my Excel spreadsheet full of my library. Library is you take a tissue sample from the cow that you're studying, you extract the genetic material, and you run it through the instrument, and you get back a pile of reads. The pile of reads for that cow is that cow's library. So we're going to pass in the size of the library for each of our columns, and that's just the total number of reads. Some of our samples, they're not going to all have the same number of reads. There's just because of differences in how you actually collect the sample and extract the information and everything. So there will be differences in the library sizes. That's important if you think about it, because suppose that cow 4, for some reason, we got more genetic material from that cow than from cow 3. But in terms of expression levels, maybe they don't differ systematically. They're all pumping out the same proteins at the same abundances. But just because we had a deeper coverage of cow number 3 than of cow number 4, it may look like there are systematic differences in the abundance levels. So it's important to keep track of this library size thing. We're also going to pass in a comparison group indicator, because there are comparison groups in this data set. There's a control and there's a treatment. And R broke, looks like. What's this? No. Unable. Okay, warning. Not writable. Had this before, and then it has not been an issue lately. Do you remember whether there was a fix for this? We're in the right working directory. The only other thing was it was the working directory. That was the problem. But we're in the right place. Try this one more time. Has anyone done this while we're sitting here? Has anyone tried installing EdgeR? Did you have any problems?
I got it. I got EdgeR. I just couldn't do the Ellen.
Okay. Would you like to use a personal library? Yeah? Let that work a little bit more. Both EdgeR and DESeq2, because they do this over-dispersed Poisson-based model, there is a requirement that we estimate the dispersion itself. If we were to say our data are Poisson, then there's only one parameter that we need to worry about. There's the mean, because the mean is the variance. Same thing, so all I need to do is estimate the mean. If we're going to make the model more flexible by allowing for over-dispersion, then we have to incorporate an additional parameter. So what we're going to be saying, EdgeR and DESeq2, is that each gene has its own distribution. It's a Poisson distribution. Each group has its own mean, the control group and the treatment group. And I'd like to do inference on the difference in the means. Just like if we were doing a t-test, let's test whether the mean expression level for treatment is the same as the mean expression level for control. You could do a t-test. We want to do that now, but we have Poisson over dispersed data. And that's not a standard problem. That's not a t-test or a linear regression model, so we have to use some custom methods. And Edge R has its custom methods piled into this function called estimate common dispersion. So what we're going to be doing is gene by gene, we say there's a Poisson distribution. There's a mean for control group, there's a mean for treatment group. And there is a separate dispersion parameter for this gene. There's two means and there's a dispersion parameter. We need to estimate all of those things before we can do inference, get a p-value. Okay, so if we were to think about, let's think about a data set full of omics information, M by N matrix of numbers. Let's just think of these as continuously distributed numbers. Let's forget about the read count thing for a second. So maybe these are gene expression microarray data. Genes in the rows, individuals or samples in the columns. What we've already done when we talked about the p-values and the false discovery rate stuff was to go row by row and do, say, a t-test or ANOVA or linear regression to get a p-value. If we were to do a t-test, let's suppose that the first half of the columns are control group, the second half of the columns are treatment group. Then we could compute a t-statistic for gene I. The t-statistic for gene I would be sample mean for gene I in, say, control group minus the sample mean for gene I in treatment group, divided by some standard error, some standard deviation estimate of that numerator. So I'm just going to put standard error. That's a general form of a test statistic. If I want to test the null hypothesis that the mean difference is equal to 0, say, then I can do a test statistic that is my estimate. I can think of it like my estimate of the parameter of interest, which in this case is mu control for gene I minus mu population mean treatment for gene I, divided by the standard error of my estimate. That's the form of a t-test. That's the form of lots of different types of tests, hypothesis tests. SEI. Let's put an estimate underneath, or an I underneath that standard error, because gene I has its own standard error that we need to put in here. In the early days, when this was all microarray stuff, t-test stuff, what would happen initially was gene by gene t-test. We've done that before, just a straight t-test for each of the genes individually without regard for the other ones, the other genes. So I would go to gene 5 and I'd get the mean for control, I'd get the mean for treatment, and I'd get a standard error estimate, get a t-test, t-statistic. What some people noticed was that these dispersion parameters, that is another way to call that is a dispersion parameter. Dispersion has to do with spread. How spread out are things? What some folks noticed was that these dispersion parameters, when we have small sample sizes like we often do with omics data, thousands of features, but a handful or a dozen or something like that, samples. The standard error, the dispersion estimate, can be highly variable. So in statistics, we have parameters. Those are the quantities in the population whose values we wish we knew, like the mean survival time of people with stage 5 prostate cancer. If I could find every person with stage 5 prostate cancer, I could compute that and we'd be done. We would know the mean, the population mean. But in statistics, we never know the truth, so we're stuck taking samples and then estimating the truth. So when we do a t-test like here, we are estimating things. That's an estimate of the mean in the control group for gene I. That's an estimate of the mean in the treatment group for gene I, and this is an estimate of their standard error. Any estimate, also called a statistic. A statistic is just a number you compute from data. Any statistic is random because the data are random. Because the data you collect come from a distribution, anything you compute on them has a distribution. That's the sampling distribution. Any statistic has a sampling distribution. So everything that I've circled in that formula, the t-statistic formula, is random and has a distribution. When you have a large sample size, you can, what we typically do in practice, practically speaking, is treat this as not all that random anymore. If I have a large sample size, treat that as if almost like it's known, and use the normal distribution as the sampling distribution for t. When you have a small sample size, that little trick is not very plausible or defensible, because with small sample sizes, standard deviations, standard errors, can be highly variable, more so than means. So, what people noticed was that by doing this, just a straight gene by gene t-statistic, we would get we'd get more noise than we want in the results. So long story short, what people started doing about 10 years ago was, instead of estimating the standard error gene by gene, take all the standard errors. So imagine here's my standard error estimate for gene 1. Like if I wanted to do a t-test for gene 1, go compute that thing. It might be like the sample standard deviation for treatment plus the standard deviation for control divided by some normalizing number. There'd be a standard error for gene 2, ... standard error for gene M. And it turns out you can improve the quality of your statistical inference if, instead of using those individually computed or estimated quantities, you put them all together somehow and turn them into smoothed, let's call it SE tilde 1, SE tilde 2, ... SE tilde M. SE tilde 1 started as just the usual standard error estimate for gene 1. That's what you would have had if you had just gone to gene 1, ignored every other row in the data set, and just computed a t-statistic based standard error. Over on the right with the tildes, we've taken all of those thousands of standard errors and we've smoothed them or shrunken them in some way. This shrinking idea is a statistical trick that kind of paradoxically can bring you improvements. When you're applying a statistical model to a random draw from a population, you want a couple of things. You would like to have an unbiased picture of the population. So if I want to study U.S. adults, I don't want to just study retirees. I'm interested in the whole population, so I want my sample to be representative of the whole, unbiased. You also want to manage the variability or the uncertainty in your results. So suppose that I could estimate the lifespan of U.S. adults on average perfectly. So if you give me enough millions of people and let me guess every time, the average will be spot on. But suppose that my variability is such that I could be plus or minus 20 years, then I'm unbiased, but I'm highly variable. On the other hand, I could always guess 80 years for everybody, in which case there's no variability in my guess, but there's a lot of bias because I'm going to get it wrong for a lot of people. What you would like to do is balance both of those, and there is a trade-off. You can be unbiased but highly variable. You can be no variability or low variability but high bias. And it turns out that there is an optimal trade-off. And in particular, sometimes you can do better than an unbiased estimate by cooking in or allowing for a little bit of bias that brings with it a correspondingly large amount of decrease in variability. So if you've ever heard of the mean squared error, that's a measure of goodness, of fit of a statistical model. And what it is the bias of your model squared plus the variance of your model. You don't want a lot of bias, but you also don't want a lot of variance. So MSE is a way to put them both together. So if you want a good statistical method, you can characterize it in different ways. You could say it's unbiased, that's good. You could characterize how variable it is. But it may be that here's a method A and here's another method B. A could be unbiased, B could be biased, but B could have a lower mean squared error than A. So in applied statistics, it's possible to trade some bias for better overall performance if you can drive down the MSE. And that's what this trick is all about. EdgeR and DESeq2 do this. They take these dispersion estimates, and whereas these are unbiased estimates, if you were to go to gene 1 and take its numbers, its numbers and compute the usual t-statistic-based standard error, that is a defensible choice of an estimate because it's unbiased. These are going to be biased. That's not good, is it? But it turns out that by doing that, you drive down this piece of the puzzle greatly. And overall, you get better inference. So nowadays, when you do omics statistical analysis, often there will be this going on behind the scenes. If you read the literature, you might see people talk about moderated test statistics. And that's what they're talking about. Moderated in the sense that we are playing with the denominator, that dispersion parameter. We're smoothing it, we're sharing information across all the genes in order to make those more stable, which results in overall more reliable estimates and inference. Okay, so that seemed to have worked. Let's see if I still need to do this. Okay, so EdgeR does its approach to estimating this dispersion parameter, and we access that algorithm here, estimate common dispersion. What EdgeR does is say there is, instead of m distinct dispersions, there's really just one. And we're going to take all of these estimates and shrink them towards that one common value. That's one way to do this technique of shrinking. We're shrinking these noisy estimates closer to something more stable. That's what EdgeR does. That's what it means by estimate common dispersion. We're going to take all of them and estimate some overall one and shrink towards it. Once you've done that, then you just call a function or two in the EdgeR case to compute p-values and get q-values. And from there, we know what to do. We have rows, thousands of genes, and now we have p-values and q-values. With DESeq2, it looks very similar. We have to set up another specialized object that DESeq2 recognizes. In this case, we use this function. And I'll show you one other thing in a second, but what we end up doing is just calling this function deseek. And when we call that, I'll show it to you, it'll spit out some interim output that says I am estimating or I'm fixing the dispersion estimates. After that, it's p-values and q-values, just like before. Okay. So I think what I will do as soon as this is done working, we need to get the data in. So I need to library, edge R, and go seek. Why so long? The other piece of deseek2 that I wanted to mention while we're waiting is this right here, rlog. In your homework, you're asked to do some exploratory stuff on the transformed read counts. So we have read counts. These are actual, 422 times gene X was overlapped by a read. Suppose we wanted to do hierarchical clustering or principal component analysis. What the authors of deseek2 recommend is that you not do that on your read counts themselves. So most of these exploratory techniques like clustering and principal components are most applicable, I guess most naturally applied to just numbers, numerical continuous data. You don't have to have continuous data to do these things, but over-dispersed count data maybe are not the most appropriate. So they have provided their own custom transformation. It's like a log transformation, but it's got some special bells and whistles. So we're going to do this before you do clustering in PCA. Turn your read counts into continuous numbers. And we may just be out of luck here. Lazy loading. Typical. Yeah, it's a good question. How do you test whether this is an appropriate method? For the most part, you would use either somebody's math that they wrote down and said, this is similar to your data, and mathematically I can show you that if you do this moderation, you get better power, statistical power, say. You pick some kind of outcome of interest like statistical power or number of genes that you can find at a specified false discovery rate, and you could try to show mathematically that you'll have greater power if you were to do this. Or simulations, computer simulations, kind of like what you're doing with your breeding experiment or your selection experiment. Let's suppose, and I'll show you one in a second actually, let's suppose we have.
So you hope if you do enough simulations, capture the iteration and understand.
Yeah, if you were to do, a series of simulations, here's a simulation with 2 comparison groups. Here's a simulation with 5 comparison groups. Here's a simulation with a small sample size. Here's a simulation with a large sample size. Try a handful of different simulations. Try the 2 things that you're going to compare. And since you're doing a simulation, you can show at the end of it, I got more stuff. I found I had higher power than this one did. So you can either try to show it analytically using math, or you can try to show it using your computer, using simulations. Warning messages, okay. Warnings are okay. Let's see if this works. Let's library these two packages in. So edgeR and goseek. Error. Package gene len database required could not be found. 95 megs. I will show you something that I tried because I was curious. How do you know this works? One way that you could check whether this is doing something sensible is to simulate some data where there's nothing going on, where there's no differences between, say, control and treatment, and apply this method. Suppose that we have 5 samples from control, 5 samples from treatment, thousands of genes, and they are read count data, so we generate them from a negative binomial distribution. There's no difference in the means between the two groups. We apply edgeR, we get p-values. If I made a histogram of the p-values, what should that histogram look like? If there is systematic stuff, if there is differential expression, we're used to seeing one of those. If there's nothing, which I could make there be nothing if I simulated data. I could simulate a bunch of negative binomial data, no differences between the two groups. I should see that. And what's a little disturbing is you don't. So I'll show you that if I can get R to cooperate. I did that. I simulated some negative binomial data. Give me some random negative binomial data where there is no difference between two comparison groups. And then do edgeR. Do edgeR. Do edgeR and do a t-test. And let's compare. I'm curious. So we'll look at that in a second if I can get this thing to finish. Okay. Let's see now. Library. Okay, maybe we don't need goseek. Let's see. Nope, I do, because that's the data set that I need. I think I can get by without that for now. Did anybody do this on your computer here? Anybody having any problems?
You didn't do goseek, did you?
I did.
I think it's required error. See, there's an error on that.
So I did install above here. I know I did that.
Well, if you saw the error and then you installed it underneath.
BioC-Lite goseek. And went through all of this. And then I libraried goseek. And it said a package that it needs is not installed.
And you installed that.
Then I installed it. Library gene len database. Let's see. Well, this works on my laptop. It's one of those fun things about bioconductor, I guess. What I think I can show you, this code, I think it should work for you if you follow the installation procedures. I think it should work for you. You run this, you'll have the data set. This code will do the differential expression analysis using edgeR. You can follow the code that follows down here, line 334 starting. This is the same stuff as above, same data set, but now we're going to use DESeq2, and that's what I'm asking you to use in your homework. So R-log transformation is right there. Once you R-log transform, if you say assay of the output, if you assay of the output of R-log, it will give you the m by n matrix of numbers. Those are no longer read counts. They've been transformed into continuous numbers. So that's how you can pull those out. In order to do differential expression, you can follow the code down here, although in the homework I referred you to this function, in binom LRT. If you do question mark in binom LRT, it'll show you how to use it. So over in ngs.r, I don't actually have that function, the negative binom LRT. Here I'm using DESeq. DESeq, that function is only for 2 comparison groups. In our case, if we have covariates or we have more than 2 comparison groups, we may want to use a regression model kind of approach. That's what that negative binom LRT does. So do question mark that function name and it'll show you the syntax. It's not hard to use. With what time we have left, let me just show you this simulation, because I don't need the data to do it, where we're going to apply edge-r and just ask, okay, is it doing something sensible? One way to ask, is it doing something sensible, is to do a simulation. So let's do a simulation. Going to generate an m by n matrix of numbers. These are going to be our read counts. So let's loop through our m genes, and m in this case hasn't been defined. Let's say m is equal to 50,000. 50,000 genes. Okay. And I'm going to simulate data that looks, in terms of structure, like the LenCap data, the data that we're trying to get access to. So in particular, I'm going to say that there are 4 control samples and there are 3 treatment samples. But these are not real read counts because I'm going to generate them randomly. And I'm generating them in such a way that I'm going to generate 7 observations for each gene. So n is going to be equal to 7. And all of them, all 7, are coming from the same distribution with the same mean and dispersion parameters. So all I'm doing is simulating under the edge-r model. Edge-r says read counts, negative binomial. Everybody has their own mean. Everybody has their own dispersion parameter. We're going to make up a mean, make up a dispersion parameter, and we're going to make it be that there's no systematic difference between control and treatment columns. So if we do that, we have a 50,000 by 7 matrix of simulated read counts. That's what it looks like. The first 6 rows. And now let's do edge-r. So I need to create the special object class, DGEList. I need to do the smoothing of the dispersion parameters. And then exact test is one of the edge-r functions that does a test. Let's do a hypothesis test, gene by gene, treatment equal to control, yes or no. And then here's the histogram of the p-values. And we know what it should look like. We simulated the data. There's no differential expression. Should be flat. So that to me, that basically is why I recommend DESeq2. If you do this same simulation with DESeq2, there are some issues that pop up, but I think I know how to fix them. I don't know how to fix this because I don't know exactly what the problem is. But this is not right. You know, it should not look like this. In particular, it's saying there's a bunch of differentially expressed genes. This peak over here, 4,000 or more genes had p-values that were really small. It should only be 5% by chance. And then also strangely, over here at the right, there's a huge pile of them that had basically a p-value of 1. If I had to guess, it has to do with lots of zeros. Too many zeros. But that's a very simple sanity check that the method should be able to pass, I would say. I mean, that if anything, it should be able to give me correct p-values. So if it were my reputation on the line, I would go here because I don't think it's as susceptible to that issue. Questions? What? Okay, pick one. Okay. The other method that's mentioned in the script here, but I'm not going to spend class time on, is lemma. Lemma came from the microarray world, and basically what you can do with lemma on NGS data is transform your read counts into something more continuous-like, kind of like the R-log transformation, and then you just do t-tests and linear regression. That's what lemma does. So it's really not set up to be specific for next-gen sequencing. It does have a few additional bells and whistles that these other techniques above do not, but I think you're safest using DESeq2. All right, I will quit there if there's not more questions. Do you have a question? Yeah.
So generally for the next-gen sequencing results, you always want to transform the data?
Let's see. So I like DESeq2, and what DESeq2 does is a couple of things. One, if you want to do exploratory stuff like clustering and PCA, I would do this R-log transformation first. Turn your read counts into something more like what you're used to, continuous-like numbers, to make pictures, clustering, PCA. When it comes to actually computing p-values, doing a hypothesis test, use the specialized methodology in DESeq2, which works straight off of the read counts. So whenever I pass something along to DESeq, I'm giving it this object, and that object we created up here using the actual read counts, not the transformed read counts. All right. Homework's due Wednesday, and we'll see you then.